So hi, I'm Paul Macklin. I'm an associate professor in intelligent systems engineering. I want to thank you all for coming today, and thank you guys for inviting me to talk. So this work is really a collaboration of a lot of great people, my lab, some collaborators, here at ISC and at IU, at Argonne National Lab, and all sorts of places. Really grateful for the collaborations from some funding from the Breast Cancer Research Foundation, the Jane Koskinas, Ted Giovannis Foundation for Health and Policy, the NCI, and the NSF. Let's see if I can find the buttons here. So let's see if we can tell a story in 5 minutes. So tissues are complex systems. They're full of cells that have single cell behavior that are influenced by their tissue microenvironments. They exchange chemical and mechanical signals to change the behaviors, communicate, and even coordinate their behaviors. And they secrete things that alter their microenvironments. And so as scientists, our goal is to build computational models that help us understand these complex systems. And as biologists, as clinicians and engineers, our goal is to actually engineer and steer the behavior of the system to tune the behavior of single cells to get a desired system outcome. And so we build computational models of these systems. Biochemical communication in tissues means 5, 10, or more PDEs, of diffusing chemical signals. Traditional solvers solve these one at a time, so it's really challenging to extrapolate them to true biological scale. So we built an open source platform that can solve 5, 10, or more of these signals in 3D just using desktop computers. It's all nice and parallelized to open source. Then on top of that, we built a system called PhysiCell that takes this basic system and adds on the cell biology. And so it's a very advanced, flexible framework. It works cross-platform on systems ranging from the Raspberry Pi to Craze, all self-contained and cross-platform. And you can extend it with things like custom data and custom functions applied on a cell-by-cell basis dynamically at runtime. And because it's open source, we get contributed functionality from other people. For example, Marie Curie recently donated functionality to add gene Boolean signaling networks to each individual cell agent. So let's just show some examples of how we use this. So for example, here, we're looking at an immune response to a tumor. And so here, the red immune cells are homing towards the tumor. Then they form mechanical adhesions to individual cells, test MHC for immunogenicity, make a decision whether to try to kill the cell, and they either succeed or they give up and then they move on. And through this basic 3D model that we were able to run, we found that spatial effects and stochastic effects have a big difference driving the success or the failure of an immune response. This is something you don't get out of an ODE model, and something you don't really get without modeling dynamics. But a single simulation is just a fun demo. So to make a useful tool, you need to be able to scale. So we worked with Argonne National Lab to distribute a large parameter space investigation on the Crate cluster, and we ran 250 simulations in like 2 days. That's a year and a half of science done in 2 days. So by scaling something up to high throughput computing, you can do big science. And so this really gives us a new paradigm, a new engineering tool to really transform the way we look at science. Now you have a simulation that can run in high throughput. So imagine you take 1,000 hypothesis sets, distribute it on 1,000 copies of your model on 1,000 compute nodes, distribute it on a supercomputer. Take your 10 best ones that match your design goals or your data, iterate, and you've got a genetic algorithm. So you can turn these things into optimization problems. What is the best set of design rules to attain a tissue objective, to design and build some tissue? What's the best treatment protocol to get the best response? What's the best set of hypotheses to match a set of data? And so there are all sorts of uses for a system like this. For example, we have a new NSF project to build nanotherapy simulators that are actually hosted in the cloud. So you can ask what-if questions. What if you can design a nanoparticle that drops a drug in low pH zones? What if you can design that drug to change the cell migration behavior? What happens? Is it worth developing? Or in synthetic biology, suppose you could program bacteria with new genes and insert new behaviors, turn on the secreted chemical signal, make cells migrate towards the signal, make them attach and detach from things. Can you build a drug transport system? Can you build a cargo delivery system? Is it worth developing? Does it work? Well, with these kinds of models, you can explore these questions in high throughput before you spend the money on the experiments. And so really the takeaways here, that we've built powerful 3-dimensional models of complex systems that account for the mechanics and the biology and the diffusion, and can be very customized and tailored, and can run them in high-throughput supercomputers, and can run all sorts of hypothesis-driven science or clinical science or engineering. It's all open source. You can download it. Anyone can try it, but it's BSD licensed, so it's commercially compatible. So And really, I think this is really uniquely IU here because we are taking unique strengths here, math, biology, high-throughput computing, high-performance computing, combining them to platforms that solve real-world problems. And it's something that we're doing here today at IU in Intelligent Systems Engineering, and I'm grateful to be a part of that. So look us up. We're online at mathcancer.org. We're on Twitter. Everything's mathcancer. Make it easy. And I'd be happy to talk with anyone of you.